Hello, Reed Samori here for our Week 5 Advanced Concepts presentation. We're talking about the process of cellular respiration and how much ATP we can net from one molecule of glucose. So I tend to be wordy, and this is a lot of information. So if it sounds like I'm pretty much reading what I wrote, that is the truth. So apologies if it's a little dry. But there's a lot of detail here, and I didn't want to miss anything. So first things first, we'll start with glycolysis, the first step of glycolysis, which is the phosphorylation, don't say that very much, phosphorylation of glucose into glucose 6-phosphate, which is catalyzed by hexokinase, which traps it in the cell. So that actually costs 1 ATP. So we're investing in the process at this point. The molecules isomerized, and then fructose 6-phosphate is also phosphorylated to fructose 1,6-biphosphate by PFK. And again, this phosphorylation process cost an ATP molecule. So we've now netted -2 ATP investment at this point. All right, now further on in the process, after we've split it into two 3-carbon molecules, glyceraldehyde 3-phosphate is oxidized in a reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase, which reduces NAD plus to NADH, and this happens with both GAP molecules. So now we have -2 ATP, but we have 2 NADH. Let's see what happens next. Phosphoglycerate kinase catalyzes the transfer of the phosphor group from 1,3-BPG to ADP, which produces 3-phosphoglycerate. And so now we've, done substrate level phosphorylation, making 2 molecules of ADP, one for each GAP molecule that we created. So now we have a 0 balance of ATP and 2 NADH. We can skip down to the last step of glycolysis and pretty much have the same thing happen, where pyruvate kinase produces pyruvate from PEP and phosphorylates ADP. And again, this happens twice. So now we're at the end of glycolysis. We had 2 ATP molecules and 2 NADH molecules. Now, before we can move on to the citric acid cycle, we have to prepare pyruvate by converting it to acetyl-CoA through oxidative decarboxylation. And at the end of this process, we get another 2 molecules of NADH when dihydrolipoamide is oxidized to regenerate lipoamide. And the electrons are transferred from FAD to NAD+. And of course, again, this happens twice because we have 2 pyruvate molecules. And our total now is 2 ATP and 4 NADH. And now we are in the citric acid cycle, where 2 carbon from the acetyl group from acetyl-CoA condenses with oxaloacetate to form citrate, and an isomer of citrate is decarboxylated twice, both of which reduce NAD plus to NADH. The first one, oxidation of isocitrate, is catalyzed by isocitrate dehydrogenase to form alpha-ketoglutarate, which is oxidized by alpha-ketoglutarate dehydrogenase, the dehydrogenase complex forming succinyl-CoA. And again, this yields 2 NADH per acetyl-CoA. That means 4. So now we have 2 ATP and 8 NADH. Let's see. All right, so now in a reaction catalyzed by succinyl-CoA synthetase, succinyl-CoA molecule has the CoA displaced by orthophosphate. A histidine residue removes the phosphoryl group and transferred it to ADP. Again, this happens twice, making 2 succinate molecules and 2 ATP molecules. So now we have 4 ATP and 8 NADH molecules. Let's see. The enzyme that catalyzed the oxidation of succinate to fumarate, succinate dehydrogenase, is a part of complex 2 on the electron transport chain. This reduces FAD to FADH2, which does not leave the complex. That's important later. So now we have 4 ATP, 8 NADH, and 2 FADH. And finally, fumarate is hydrated to form malate. The oxidation of malate is catalyzed by malate dehydrogenase, which ends the cycle and reduces NAD plus to NADH, netting 4 ATP, 10 NADH, and 2 FADH total. So that's what we have to work with as we move into the electron transport chain, where we can harvest these high transfer potential electrons from NADH and FADH2. Ultimately to create a proton gradient, which powers oxidative phosphorylation of ADP via the ATPC synthase enzyme. Complexes 1, 3, and 4 each contribute to the gradient by pumping 10 protons per pair of electrons out of the mitochondrial matrix into the intermembrane space. Complexes 1 and 3 pump 4 protons, and complex 4 is 2 per pair of electrons. And so ATP synthase has subunits in the C ring. That's the part of the enzyme that moves the rotor. And each of them have to be loaded with a proton. So an H plus ion. And we'll say 8 to 10 gets us a round number of 3 ATP molecules. So I think it depends on the organism, but most people seem to agree that a full rotation of the C ring leads to the synthesis of 3 ATP molecules, and so anywhere from 8 to 10 rounds to 3. And per the book, it takes an additional proton to get ATP into the cytoplasm. I've read other things that don't agree with that, but most people seem to agree that it takes 4 protons to get 1 ATP molecule. And now we can take that information to actually do the math to get to 30, which is what we were asked to do. So again, just to go over our totals again, I know it was quick, but we have 4 ATP, 10 NADH, and 2 FADH2 molecules, which are on complex 2. So the 10 NADH molecules give us 10 pairs of electrons. And so so that's 10 times 10, it's 100 H plus ions into the proton gradient. And so we know that it takes 4 protons to get us an ATP, so that's easy math. 100 protons gets us 25 ATP molecules. And we have to treat the FADH2 a little different because they bypass the first complex, which is responsible for 4 of the protons being pumped into the gradient, into the intermembrane space. So that means anything that bypasses the first complex only contributes 6 protons to the gradient, which is 1.5 ATP molecules. And we have 2 of those, so that's 3. Or the math, you could look at it this way, 12 protons divided by 4, because it's 3. So we have 4 ATP already produced, and then we have 25 from the NADH, so that's 29, and then 3 from FADH2, which gives us 32, which is not the number that we were asked to get. So we have to consider something else, which is how we shuttle the NADH from glycolysis into the electron transport chain, into the mitochondria for the electron for use in the electron transport chain. So there's two shuttling systems that the body uses: the glycerol 2-phosphate shuttle or the malate aspartate shuttle. And when the glycerol 2-phosphate shuttle is used, like in skeletal muscles, the electrons enter the electron transport train through Q. And again, just like FADH2, this bypasses the first complex. So instead of being the 2.5 number, we have the 1.5 number, meaning that we generate 6 protons divided by 4, as opposed to 10 divided by 4. So if we update our math now, assuming that we are doing this in skeletal muscle, we go down from, we'll just say we go down from, maybe we could just treat it like FADH molecules. So we could do the math by saying now we have 8 NADH molecules, 4 ATP, and 4 NADH or FADH2. That would work, or we just subtract 2 to get 30. So the malate aspartate shuttle is in heart and liver cells, so I don't think we can ignore it. So I think all in all, I think it's fair to say that one glucose molecule nets us 30 to 32 molecules of ATP in the body, depending on what cell we're in. And hopefully, that was 12 minutes long. That's crazy. Good night.